So we are going to know how genes are regulated. So there are large number of genes in the body. So around 20,000 to 25,000 it is expected. These are the genes which are present in human body. Now how these genes are regulated is explained by a concept known as Operon system or Operon concept. This was given in 1961 by Francois Jacob and Jacques Monod. Okay, so we shortly or in short form we say it as Jacob and Monod model of Operon system. Now what is actually a Operon? So you can say that it is Operon is a cluster of tightly regulated genes. Okay, and how this regulation happens, we will be talking about it. So when we talk about Operon and its regulation, so we have got two kinds of regulation. So either this could be a positive regulation, or even this can be negative regulation. So when we talk about positive regulation, this is known as inducible operons, and when we talk about negative operon, this is known as repressible operons. Okay, so we will be talking about inducible operon because as per NCERT class 12 CBSE, we have only one operon listed, that is Lac Operon. Now Lac stands for lactose. Okay, actually it could be even allolactose. Now these molecules, they are the inducer. Okay, now what and how this operon works, we will be talking about it. So when we talk about lactose operon or Lac Operon in short, Lac Operon, we need to draw the diagram that is given in the book, and we'll be using blue just to denote that whatever we are drawing is DNA. It's a segment of DNA. Okay, it's a hypothetical diagram. We'll make, it's a long stretch of DNA. Okay, here. So what this shows that the genes can be located side by side, or it can be located a bit far from each other. Now what are the genes that are present in the operon? So we have the regulatory sequence, sequence or genes. This is what is known as the repressor gene in the body. So this is the I gene. Okay, and then we have the promoter. Okay, then we have the operator. Okay, and then we are going to have the structural genes. So these would be the structural genes, structural, structural genes. So what are these structural genes? So these structural genes are something that needs to be expressed. So we have three genes, one after another. So one is Lac Z, then Lac Y, and another is Lac A. So we'll just write it here. This is Lac Z or Z, then this is Lac Y, and this is Lac A. Three structural genes are needed. Now when the organism is metabolizing lactose, all these genes are functional and they will produce their respective mRNA. So Lac Z will produce the Lac Z mRNA, Y will produce the Lac Y mRNA, and A will produce the Lac A mRNA. Now this mRNA will go for translation Okay, all of them in fact will go for translation, their own translation. It may create confusion. We'll just show it separately. So this will go for translation, even this will go for translation. Okay, and this will go for translation. And they will produce their own proteins. So this is the folded protein. So the name of this protein is beta galactosidase. Okay, and this one is going to produce a protein which is permease, and this one is going to produce a protein which is unrelated in the lactose metabolism, and this is known as trans-acetylase. Okay, so now what happens is, when bacteria is in a medium and there is no lactose, so it doesn't need the expression of these genes. So what happens? The lactose operon has to be shut off. So here we see that the repressor gene, this keeps on producing the repressor mRNA. So mRNA of the repressor, or repressor mRNA, and this repressor mRNA goes for translation, and this is going to produce an enzyme or active repressor protein. And this active repressor protein will go, what is the job of this? That it will go and it will block the operator. Now when the operator is blocked, the promoter site RNA polymerase cannot land. So it will not be able to land in the promoter site, and the transcription followed by translation of these genes will not occur. So even this will not be present, this will not be present, and this will not be present. So what has happened? The operon is shut off. Operon is off by default. To start with, we'll say that the operon is off. Now, so this was case 1. In case 2, what happens is lactose is added onto the medium. So the bacteria is present here and somebody is adding lactose. Then what happens? So some of the molecules of lactose, they will go for passive diffusion. Okay, and they will bind with the active repressor. And when lactose molecule, they bind with the active repressor, what happens is this active repressor becomes inactive. Okay, so this will now look like this. It has become inactive, won't be able to attach to the operator. Okay, so it will fall off. As a result, what is going to happen? This repressor will not be present to hold on to the operator. Okay, so operator क्या हो जाएगा? Operator is free. So now what happens? RNA polymerase can actually come and transcribe. So RNA polymerase will come and it will start the transcription and translation of all these genes. So I'm just putting up the cross. I'll just redraw it. So here transcription will start, and followed by the translation will start. Okay, and all ticks, all check mark, it will start, this will start, and these proteins will also be formed. Again, I'm writing the name of these proteins. What is it? Beta galactosidase. Then this is permease, and this is trans-acetylase. Okay, now they can ask you a question: what is the function of these proteins. Okay, so beta galactosidase will help to metabolize lactose. Okay, so we can just write it like this. So we have lactose, and then this is beta galactosidase. Okay, and this will break lactose into glucose and galactose. Okay, so you should know that lactose is disaccharide. Okay, and glucose and galactose, these are monosaccharides. That means they are made up of only one unit of sugar. Okay, what will permease do? So permease, this will create, or it will create transient pores in the cell wall of the bacterium or the bacterial cell, and that will allow more lactose to diffusion, more diffusion of lactose. Okay, and transacetylase, so this is unrelated enzyme, unrelated to Lac Operon. It doesn't function in the this metabolic pathway, but the gene is side by side, so it gets expressed. So unrelated to Lac Operon helps in transfer of acetyl groups from one compound to another, one compound to another. Okay, and you should know from where these genes are coming. Beta galactosidase, this is coming from LAC-Z gene. Okay, permease, this is coming from LAC-Y gene, and transacetylase, this is coming from LAC-A gene. Okay, I hope you're getting this. And once the bacteria has metabolized all the lactose, then what is going to happen? The lactose will be scarce in the medium, and it cannot go and bind to the repressor. So now the repressor, what happens is, becomes active again, and then it will again go and block the operator. Okay, once the lactose in the medium is used up. Once the lactose in the medium is used up, operator is blocked, RNA polymerase cannot do its job. So again, the transcription followed by translation of all these enzymes are halted. So no lactose in the medium, the bacteria doesn't need to produce these gene products. Okay, so I hope this clears your concept, and let me know what more topics do you want to get videos on, and I will make short videos on these topics. Thank you.